So, John, as just discussed, we, maybe need to produce a new vaccine in a rapid response to an outbreak. But how can we rapidly deploy these and make sure that they're safe?
As most people would know, the typical timeline for getting approval for a human vaccine in general takes many years of human clinical trials, and it can take 5 or 6 years to get approval for a new vaccine to be put onto the market and used in humans. Clearly, that's unacceptable and simply does not work in the face of pandemic outbreaks, where you might need a new vaccine for a new strain of virus as quickly as possible. One of the reasons for the long time frame for getting approval for a vaccine is that they very carefully monitor the safety aspect of it, and if there's any adverse effects, it's not allowed to progress, or it has to be remedied. You can't really take that kind of approach with a disease where you have up to 90% mortality and you need to act very, very quickly. So there has to be very much an element of risk-benefit. Okay, it might not be a perfect vaccine. There might be some side effects, but dying from the disease is ultimately much worse. And so ultimately you're better off employing a vaccine earlier than it would normally do in a commercial setting. And one way around that is that there's lots of efforts now being put into how these vaccines are manufactured and delivered. And a lot of them are being produced in what's called viral vectors. So you would take a virus that's normally not dangerous to humans. It might be an adapted adenovirus, for example, and you can put certain genes for an Ebola virus in that, administer it to a person, and the person would develop immunity to Ebola. If it was accepted that the backbone or the vector is relatively safe in humans, you would be able to get much faster approval of adapting that to different zoonotic outbreaks, because a lot of the safety issues do with the viral vector, not so much the antigens that you put in there that you want to get protection against.